Hello, everyone. This is Xing Wang. I want to take this opportunity to share some of our recent research with you. The title of my talk is Virus Sensing and Inhibition with DNA Star Strategy. Okay, so as you know, most epidemics and pandemics happened in recent years were caused by virus. Okay. However, after so many years contact with the virus, where still lack of the effective diagnosis, point of care test, and the cures for virus diagnosis and the treatment. So that's basically the case for the ongoing COVID-19 pandemics. So these challenges basically are the driving force behind our research working on the virus sensing and the inhibition. Before I talk about the details of this research, I want to thank the three major heroes behind the curtain. Paul Kwong was the leading author of this work, and it's the undergraduate research intern in my lab when he was still at GHU. Shaokang Ren is a graduate student from my research collaborator, Dr. Jie Shao's lab in Nanjing, China. And Megan Kaiser is my graduate student who is now at MIT being a postdoc. All right, so the platform or strategy we're developing here is taking advantage of the polyvalent pattern matching strategy. That's the new thing we developed in this work. So to demonstrate this strategy on virus sensing and inhibition, we picked dengue virus as a representative. The major reason for that is dengue displays the most complex epitope spatial patterns among all the known viruses. So we basically analyze the cryo-EM structure of the dengue virus, and the red dots, many over here shows the envelope protein domain 3 or ED3 throughout this talk. That's the major launching point of the neutralizing antibody of the dengue virus. So based on this pattern, we further simplify the special distributions of ED3 into two types of clusters. One is a 5-member cluster, the other 3-member cluster. When you center the 5-member cluster in the middle and linked the neighboring clusters together, it will form a star shape. That's basically the starting point of our DNA nanostructure design. We call it the DNA star. It contains a pentagon in the middle and 5 points surrounding that. So we have made two forms of the DNA star. One is a compact form, the other fully stretched. As you can see, when the DNA star is fully stretched, it will match the spatial pattern and spacing between the ED3 clusters. I will talk about why we have a compact form of the DNA star over here in a bit. So in order to enable the interaction of the DNA star scaffold with the dengue virus, we need to decorate this DNA star scaffold with the binders or ligand that you can interact with the ED3. So over here in this study, we use a known aptamers involved by others that can specifically interact with the dengue virus ED3. So the other groups of the molecules we're using currently to target other viruses are small molecules. So I'm not going to talk about that over here, but I want to point it out. This is another option. So basically, we put a 10 ED3 targeting aptamers on each of the vertices of the DNA star to make it a nanostructured aptamer patterns that can potentially match the spatial pattern and the spacing of the ED3 on the dengue virus. On the right, we did an AFM to show the successful formation of the DNA star. So in order to verify our hypothesis, which is the fully pattern matching, that's the essential element for the maximal binding affinity and specificity, we use SPR, which is a high throughput approach to study the binding affinity. That's one of the atoms it can study. It can study other things as well. So to do that, we immobilize the active dengue virus on the SPR chip and flow different DNA constructs with the different amount of the aptamers through the chip and study their binding affinity. Over here, we made the partial DNA stars continue 1, 2, 3, 4 triangles and a full star and flow through the SPR chip to compare their binding affinity. So we use the monovalent aptamer as a baseline. You can see that the binding affinity jumps about almost 1,000-fold compared to the more than 1,000-fold actually compared to the monovalent aptamers, and also with the increased number of the triangles matching the full star pattern of the ED3 domain, will get the maximal binding affinity. Okay, this basically verified that we didn't overcure in this pattern matching scenarios. Okay, so the other missing element we need to get in is the signal readout mechanisms. So over here, we borrow the concept of the molecular beacon. So we have as I mentioned a moment ago, we have 5 stem loops in the middle of the pentagon shape of the DNA star, and each of the loop is flanked by a pair of the fluorophore and quenchers. So that in the presence of the dengue virus particle, the aptamer will interact with the individual clusters. In order to do that, they need to stretch this stem loop and then in turn separate the fluorophore from quencher to give the fluorescent readout. Okay, this is the actual diagram of our dengue sensing setup. So basically you have a DNA star with the 10 aptamers in a compact mode, and 5 pairs of the fluorophore and quencher, they're physically together, so the fluorescent is quenched. In the presence of the dengue virus, which basically will give you match the ED3 pattern, then this DNA star will start to stretch itself, the 5 loops in the middle, and then separate the fluorophore from quencher and give you the fluorescent readout. So in order to test the limit of detection of this DNA star sensor, we expect the active virus in human serum or human plasma. So as you can see from this plot, the limit of detection of a DNA star can go as low as 100 PFU per ml in the human serum sample. So I want to point it out that since the DNA star and the dengue virus can interact pretty quickly, that's partially due to the nature of the aptamer interaction with their target, a protein target. So we can easily get the sensor sensing result in 1 to 2 minutes. The cost of this processes, including all the DNA materials and the plastic wares, is about 15 cents per test. Okay, so what's the meaning of these numbers? I will talk about in a few minutes. So this diagram shows the we plotted our DNA star sensor in the diagram of all other gold standard diagnosis method. The 2 very close one in terms of the sensitivity is RNA detection based on the RT-PCR. Virus isolation is also very sensitive, but it takes a long time, weeks, in order to culture the virus to identify the identity of the virus. So we're not comparing that in this comparison. And the other one is the antigen test using the ELISA kit. So in order to exclusively conclude that our limit of detection is better than RT-PCR, we collaborated with a researcher who has lots of experience in doing RT-PCR against the patient patient samples containing either dengue virus or other types of virus. We want to make sure our comparison is highly conclusive. So based on her 6 trials of the RT-PCR study using the human serum, human blood serum SPECT virus samples, the limit of detection of that is 100 PFU per ml. The ELISA-based analysis, ELISA-based diagnosis is known to have a low sensitivity, so I'm not going to spend too much time over here. Instead of showing that the cost and the time in order to get the result. So I want to spend a few sentences on the RT-PCR sensitivity comparison to our DNA star sensor. The reason why RT-PCR, which is still the gold standard method for pathogen diagnosis, has a worse sensitivity compared to our DNA star sensor that can directly detect the intact virus particle is that. So is that each of the virus particle only has one RNA genome. So in order to do the RT-PCR, you have to do the virus isolation, lysis, RNA extraction, RT, reverse transcription, and the PCR. During these processes, you are easily losing lots of RNA molecules. However, for our direct virus sensing through the epitopes on the virus surface, is that each of the virus, they contains more than one, much more than one copies of the epitopes we can target. So in that sense, we don't need to, we have a more target to detect, as well as we don't lose any of those materials during these processes. Okay, so we made a table to compare our method with other gold standard method in terms of the cost and the time to read and the meaning of this sensitivity. This will really provide a unique approach for early detection of the dengue virus as well as other viruses if we can expand this platform to other viruses. In terms of the cost, it's low compared to RT-PCR and the ELISA-based. It can be easily converted to point of care tests in low resources or poor environment. Okay, without using any sophisticated laboratory setup. Okay, so we basically further made a few other DNA scaffolds to further certify that this polyvalent pattern matching, that's the whole reason behind this high binding affinity and diagnosis sensitivity. So in order to do that, we made a few other DNA structures. The bivalent containing two DNA aptamers. The flexible linear molecules containing 6 aptamers and 6-point star and 7-point star. As you can see over here on this plot, the 5-point DNA star and 6-point DNA star still gives the most sensitive sensing result compared to the linear one, which is much less, and the 6-point star is almost the background, and the bivalent. I will talk about the reason why the 7-point star we think is even worse than linear sensor. So, but before that, I want to explain why the 6-point star can also give us a reasonably good sensitivity in terms of the dengue virus sensing diagnosis. The reason for that is if we rotate the virus particle and center the three-member ED3 cluster in the middle, and then you can link by linking the neighboring clusters, you will get a 6-point star shape. So that's basically another positive control to support our polyvalent pattern matching strategy. Okay, so encouraged by the diagnosis data, we go ahead, we moved ahead with the viral inhibition assays. Just use the vanilla DNA without any modifications. I know these DNA molecules can be chemically modified with the improved stability in the biological system, but we just go ahead with these vanilla DNA made structures. So as you can see, we did the plaque assay and EC50 measurements, and you can see that the DNA star, 5-point star and 6-point star, it shows very impressive EC50 compared to the monovalent aptamers. It has at least it has about 7,500 folds improvement in terms of the potency of the antiviral inhibition effect. Okay, so we did calculate the dosage cost for the DNA star, which is about $8 to $10 to get the dosage to repress or inhibit the virus infections. Okay, so over here, I want to use this simple diagram to explain why the 7-point star has a worse diagnosis sensitivity as well as the inhibition potency compared to the linear molecules. As you can see that if we attach one 7-point star onto the virus particle surface, the other 6 edges will block the access of another 7-point star to the virus particle surface. However, for the linear molecules, since it's very flexible, occupying one edge won't basically interfere with other edges interactions with the virus particle surface. Of course, if we have very rigid pattern matching scaffold, that is a 5-point star or 6-point star, depending on how you rotate it or how you draw the pattern of the ED3 clusters, it's going to give you the maximal effect. Binding affinity that will leads to the highest binding, highest diagnosis sensitivity and the viral inhibition effect. All right. So at the end, we did some live image to observe how the virus particle with and without DNA star bond behaves. So basically on the top panel, it shows the virus particle itself. It can enter the host cells easily over the time course of 1 hour. Once the DNA star is bound to the virus particle, they are not able to enter the cell, the host cell we are using over here through the live image observations. So the whole observation is happening in the serum containing solutions. So this is another zoom in time-lapsed images or the movies showing that the virus particle, once it's bound to the DNA star, it will be light up as a green color. That's basically the FAM color, the emission of the FAM fluorifer, and is not going to enter the host cells compared to the virus particle itself without DNA star bond. All right, so I will use these slides to wrap up my talk, which is the projection of this platform. We call it pattern recognition enabled virus sensing or pathogen sensing and the therapeutics, or in short, PEST. So basically, we're able to analyze the surface or the epitope patterns of a given virus or other pathogens, and then we can either evolve or find the existing ligands that can target that, and then we can design a DNA nanostructure matching or mirror that surface pattern, and we can make a patch for both diagnosis and treatment. So small molecule is a very promising ligand just because of this low cost and the small volume it will occupy in the space. The other thing I want to point out is that using these ligands, especially aptamer, we can evolve aptamers that can target different versions of the same virus, and then we can distinguish them using this pattern matching approach, which cannot be distinguished by RT-PCR, which is a sequence-based diagnosis. At the end, I want to thank you for your attention. And if you have any question, please email me. All right, thank you.